Okay, so I've started recording. If
I
you
think
don't
you
want...
started it. Yeah, exactly.
Yes, thank
This one.
you.
Got it. And then I'm just going to share my slides.
And then, Rammah, do you want Alexander or Lorena to interrupt you while you...
If you want to, of
first
course.
you want to do the
No,
presentation?
if you want to interrupt, feel free to do so.
Okay.
Absolutely no problem.
Because he tried already to make a project and maybe he has some very concrete questions.
Okay, absolutely no problem. Do you see my slides?
Yes.
Yeah. Yes.
Great. Yes. So as sorry, I have a bit of help, but that should be okay. But as Dana mentioned, so today I'll be walking you guys through QuPath and how you can use QuPath to get some information out of those TMAs. So if you, for example, want to look at the number of P53 positive cells, you can use QuPath for those purposes. So today I'm not really going to give you too much background information about the actual software itself, but it's more so to give you a demonstration of how to use it. And as Dana mentioned, if you want to interrupt me and have any questions, please feel free to do so between the presentation. But yeah, so QuPath is a software that was created by Peter Bankhead. So I'm going to turn on my laser. Yeah, created by Peter Bankhead. And I think originally what he said was when he created this software, it wasn't necessarily to replace other bioimaging softwares that are available online today, like Fiji, like ImageJ. You might have worked with some of those earlier, but the purpose of QuPath was to kind of do things to find solutions for bioimage analysis problems that couldn't be answered with Fiji or with ImageJ or those other softwares. So what QuPath is very good at doing is it's very good at working with images from slide scanners. So you have these big images that have, a lot of data, a lot of files and multiple resolutions. And QuPath allows you to open those files in a very straightforward way. So it kind of looks something like this when you open a file. And as mentioned, basically you work with large images stored at multiple resolutions, and it's kind of like Google Maps. So when you go to Google Maps, if you were to pan out, so you zoom out of the image, you get the overview of the entire image, right? But then when you want to zoom in and you want to look at a street name, for example, that same image will be shown to you at a higher resolution, but you get a smaller field of view, right? So the resolution adjusts depending on how you zoom maps, and QuPath is quite similar to that. If you pan out, you get the overview of the entire image. If you pan in or zoom in, you get a smaller field of view, but a higher resolution. So it's basically a very efficient way of opening big files. And yeah, the general idea is that if you have an image, those images are essentially filled with, or they have many pixels, and pixels are essentially billions of numbers, right? And you can use these pixels to then identify different objects. You can, for example, use those pixels to identify cells. You can use them to identify nerves, blood vessels, any objects of interest that you're looking into in your project. And once you've identified those objects, you can then query those objects. You can probe, for example, how many cells within my image express P53 or express CD8 or so on, or how many blood vessels do I have in my image, how many collagen fibers do I have in my image. So you can essentially use QuPath to query all these different objects depending on your research question. And it has very innovative tools inbuilt within the software to allow you to do so. So without further ado, I'll just get straight into it and show you how to create a project and how to upload those images into that project. This is something that you've probably done, Alexander, but just for the purpose of demonstration, I will repeat it as well. So I'm just going to go into QuPath. 1 second. I don't think you can see my QuPath right now, but 1 second. Oh. Okay, what can you see right now? Is it my PowerPoint slides or is it QuPath?
PowerPoint. It's still your PowerPoint, yeah.
It's still the PowerPoint. Oh, okay.
Yeah.
1 second.
But we could see it with your notes.
Yeah, let me share.
Now?
Perfect. Yeah. Yes. So when you open QuPath, it looks something like this. And the first thing you do, as mentioned, is to create a project. So theoretically, you could probably upload images directly onto here. You don't have to create a project, but the benefit of creating a project is that if you close QuPath, reopen QuPath, and then open the project, all of the images that you have stored within that project are open automatically. So it's always a good idea to create a project repository and store all of your images within that project. And to do so, you go into the File tab, you go into Projects and Create Projects. So I've created a specific directory for this on the QuPath tutorial and tutorial projects. It's just what I've chosen to where to put this project and then select folder. So now you have your project. And once you've created your project, you can then upload images onto that project. So I'm just going to find the images that will be today for demonstration. I'm just going to upload it onto the project. So you can do this by either the file and clicking on open and then opening the images like this, or alternatively, you could also just drag the files onto the project like that. And when you do that, drag the files onto the project, this pop-up box will show up. And basically, you can select the image provider. I usually just allow this to be default because QuPath is quite good at selecting the correct image provider. Then depending on what kind of image you have to set the image type. So for example, if you have hematoxylin image, then you would select that. But the images that we'll be working with today is so we've done immunohistochemistry for CD8 for cytotoxic T cells in a vulvar cancer cohort. So it's an IHC image, and therefore I would select H, hematoxylin to stain nuclei, and that to visualize the CD8 expression of CD8 surface marker within the cells. So this would be the correct option for this particular image. But then again, if you're working with H&E images like that I mentioned, then you'd maybe select that. But here I'll select this. Then you can also rotate the image. So this will simply just show, this will just kind of rotate the image as it sounds. But if you don't do this now, you could also do this afterwards. But like while I'm doing it now, because then every time I open the image, I don't have to manually rotate it again. But this is only if you know the layout of your image. So I could, for example, leave it as no rotation, and then I can adjust that afterwards. And then the auto-generate pyramids, because of the slide scanner file, the nature of the file itself, I tend to just leave this toggle on. I wouldn't deselect that right now, at least. But yes, so then you can just import the image, and it'll look something like this. So the image pops up here to the left, in the left panel, and you can then double-click it. And if I was to look at my Excel file, it shouldn't, I mean, because you want to match where the particular cores are according to your Excel file, so you know which cores belong to which patient, right? So this is quite hard to see right now, and I should be rotating this this image 90 degrees clockwise. So I could do that by going on View and then Rotate Image, and then you could just rotate it like this. So this now matches what I have in my Excel file. Yes. The second thing you could do is also look at the image properties. So if you click on the tab titled Image, you will see information regarding the pixels. You will also see information regarding the image type. So it's always good to like double check that you have the right image type. So if you have an HTAB image, you want to make sure that that correlates with what's set in the panel. And it also gives you information regarding the different stain vectors. So these stain vectors are essentially a characterization of what QuPath identifies as hematoxylin blue or as DAB brown. So if these numbers are completely wrong, then you won't be able to detect the white cells because the detection is based on hematoxylin blue of the nuclei. And same thing with the surface marker. If these numbers are completely wrong, you won't be detecting correct surface markers. So you can then manipulate these parameters to make sure that the software picks up the cells correctly. But I'll show that afterwards. Yes, so I'm just going to go back to PowerPoint. So now we've created a project, we've uploaded our images. The next step is, as mentioned, to set the staining vectors. So I'm just going to QuPath, but essentially, as mentioned, the staining vectors are what tells QuPath how to identify what the blue nuclei of the cells are and what the brown surface marker CD8 in this particular case is as well. So if I go into QuPath, yes?
Sorry, we are still seeing your QuPath, not the PowerPoint.
Okay. This is okay. Let me see if I can sort that out. I don't know why it's doing that. Maybe now it could be better. Now, do you see the slides? Yes? Okay, perfect. Yeah, as mentioned, we'll be looking at the staining vectors right now. So I'll just show you how to do that within QuPath. So I just have to go back into QuPath. And now I hope that you see QuPath. If not, let me know. But yes, so within QuPath, you have the staining vectors here in the left panel. So if you're already familiar with the concept of color deconvolution and you know what stain vectors you want to manually put in, you can do so. So you don't necessarily have to do it the way you want to do it, but you could just double click on the hematoxylin tab here, for example. This will show up, and then you can set manually the vector stain that you want to be using. But this is only for somebody who really has something in mind. If not, if you don't have something in mind, then you can use QuPath. QuPath will help you do this. But essentially, you can set the red, green, and blue value manually if you want to do so. And what's quite important to remember is if you're working with, let's say, 4 or 5 slides that are, the same marker. So as mentioned here, we're looking at CD8 for cytotoxic T cells, and these are highlighted in brown, right? If I set a stain vector here, and then I move on to the next slide. So now I'm at slide 1. If I then go on to slide 2, I want to try to maintain the same stain vector for slide 2 as well, simply because you want to acquire quite homogeneous results. If you have very different stain vectors between slide 1 and slide 2, you might get results that are not homogeneous. So it's good to copy this and then paste it into slide 2 as well. But as mentioned, me personally, I'm not very familiar with the color deconvolution. So what I like to do is I like to use QuPath to help me estimate the staining vectors. There's a function for this. And basically, what Peter Bankhead recommended is you try to find a region that has a good representation of all of the different properties that you're looking at. So you want to have an image that has a significant amount of background, a significant amount of your tumor cells, but also your stromal cells, and also cells that are unstained, so only stained for hematoxylin and blue, and cells that are stained in brown for the cytotoxic T cells you need. So basically, you just manually draw an annotation. I'll show you how to do so. Just gonna find the region that I like. Let's say here, for example. So then I will use the rectangular tool on the top left corner to just draw an annotation that kind of covers all of those different aspects I just mentioned in the image. So I have my background, I have my tumor cells, I have the stromal cells, and I also have the brown stained and unstained cells. So once I've created that annotation, I can then use a function called estimate stain vectors on QuPath to then give me some information about what potential staining vectors I should be using for my analysis. So one way to do this is to find this function, to find it manually, but I usually don't do this. I just wanted to show you myself for the purpose of demonstration. Let me see. No. Okay. Yeah. Okay. No. An easier way of doing it is just simply using the command list. So to open up the command list, you hold Control on your keyboard and then you tap L. So Control L. This will open up the command list, and here you can type in exactly what you want to do. So as mentioned, I want to do estimate stain vectors. So I write in estimate and automatically pops up. I can then double click on this, and then it will ask me this question over here. And basically, if in your annotation you include the background, it's always good to select yes here. So Peter Bankhead in his tutorial recommended just including the background and selecting yes when this pop-up box shows up. So I click on yes, and you then get this cone, or you get these scatter plots that are shown in a format. So what Peter Bankhead recommended then was that you try to make sure the lines are just on the outside of the cone. So you can try to do this manually, but it could be quite tedious because again, these are supposed to be in 3D, but they're shown in 2D. So when I move these lines are also affected. So I can try to do it manually. An alternative is simply to use the auto button in QuPath. And if you click on auto, QuPath will do this for you. And when it does so, it will also update the staining vectors for hematoxylin on 4DAB. So these are the original ones, and these are the updated ones based on the annotation that I've created within QuPath. So this to me looks okay. Maybe I'll drag this a bit down, maybe this a bit up. But you can play around with these, and then you can do your analysis and see if you're happy with it. If you're not happy with it, you can always go back and play around with these parameters a bit more. In terms of the ignore strema, this has to do with outliers. So if you increase the strema, second. Yeah, so if you increase the strema, then it will include more outliers. parameters, but I usually tend to just keep it at 1%, and it does a pretty good job doing so. So I will now click on OK, and this will automatically update the parameters for hepatoxylin and DAB in the image. And I will just keep the name of that the same, HDAB estimated, based on the estimated stain vector function within QuPath. And then these numbers are updated automatically. And as mentioned, once you have these numbers, let's say you're happy with them when you do downstream analysis, I would then copy paste these numbers. So for example, I will double click, highlight these RGB values, the red, green, blue values, copy paste them. And then I would go on to my next image. So let's say I upload image 2. Import. And then in image 2, I would then manually correct these so that the same as in slide 1, right? So I just control paste the numbers that I already have from before. So I'm going to go back into slide 1. Great. So now that you have your stain vectors, you can move on to the next step. So I don't need this object anymore. I'm going to delete this. So I will now be going back into the PowerPoint. So once you've created your stain vectors, you can now do the annotation of the objects. So as mentioned, this depends on what your research question is, what it is exactly you want to look into. If, for example, you want to look into the expression of cells in both epithelial and stromal compartments, you then have to separate between the two compartments, right? So you need to manually annotate the epithelial compartment. But you also need to manually annotate the stromal compartment. And QuPath has a fair share of innovative tools for doing this. So now I'll just show you how you can use these tools within QuPath. Second. Yes. So for example, let's say I want to annotate the epithelial compartment within this core, right? So you can obviously use the same rectangular tool that I use for estimating the stain vectors to do this to a certain degree, but this tool is quite rigid, right? If I want to get those curves within my annotation, it's quite difficult to do so if I was just to use the rectangular. So I would not be using that for this particular purpose. You also have an elliptical tool that you can kind of use to annotate various stuff. But again, as you see, this is the ideal tool for this particular annotation because it's hard to separate between the two different compartments. You also have the polygonal tool. So here you essentially click around the outskirts of the annotation that you're interested in. So I can, for example, click here, click there, and so on. Click my way throughout the entire annotation. And this could theoretically work, but I don't find it to be the easiest way of doing this. So I'm just going to delete that annotation as well. Then you have the wand function. So the wand function is this one. And again, a lot of these functions, so based on, for example, let's say the wand function, you can click on it here, but you can also just click on W keyboard and it will activate the same tool within QuPath. So I can click manually, and then I can annotate using the wand function. If I then want to stop using the wand function, but I want to be able to navigate within my image, if I click there, I'm still using the wand function, right? Therefore, I have to use the keyboard button M. So M is just to navigate between the image. So if I then toggle M, the mouse changes and I can kind of move around within my image. I can then go back into my wand function by clicking W on the keyboard to continue doing my annotation, right? So I'm going to delete these annotations.
Just a moment. So Alexander, you will be doing this. You have to annotate, meaning you have to delineate all the epithelial islands within the core. So be very attentive now how Rammah will show you how to do it and also how to link the two cores that belong to the same patient.
Yes. So my favorite tool to use within QuPath is probably the brush function. So brush, you would use the keyboard command B. So you just click on B on the keyboard and use the brush function. So kind of looks something like this. But if you pan out, the brush stroke becomes even bigger. So you can potentially cover even more area. If I pan out, you can make it even bigger. Let's say I was to use the brush function, but I accidentally start brushing into the stromal compartment, and this is a mistake. I don't want to do this. You can then erase that annotation or part of that annotation by holding in Alt in the keyboard and then erasing the part that you've created that shouldn't be annotated. So you can do that as well. But let's say, yes, I'm going to annotate the epithelial compartment, right? So I'm using the part function to do so. I'm going to do a very rough outline of this just for demonstration. But let's say something like this. There. And then something like that. Let's say. And let's say this shouldn't be part of the annotation. I can then, as mentioned, hold down Alt and erase this part. And I can include this in the annotation. So maybe something like this. But let's say I also want to include this in my epithelial compartment. So I can then use, the problem is if you were to use the brush function to create a new object here, QuPath will identify these as two separate objects, right? You don't, like if I highlight this one's highlighted by itself. If I highlight the other one, this one's highlighted by itself. So one way of solving that is obviously you can highlight this one, hold down Alt, click on this one, then both are highlighted. Then you could potentially merge those two. So let's see if I could edit multiple and merge selected. So this will now merge them. If I deselect the object and select it again, you'll see that both objects are now selected, right? An alternative is obviously I could just erase this part. I could just draw a straight line and annotate this object as well. And then I just erase everything. So whatever is easiest for you to do. I find this quite straightforward to do, in my opinion, but yeah, you just have to figure out what works for you, right? So after I finish this annotation, I can set class. So setting class essentially means that I'm gonna specify that this is the epithelial or tumoral compartment. But as Nana mentioned, you want to look at the expression of your marker in both epithelial and stromal compartments. So one way of doing it is that similar to how I annotated epithelium, you could also annotate the stroma. So you can kind of be careful and annotate around the stroma and just kind of try to make it as accurate as possible. But thankfully, QuPath has a pretty innovative function where you could, like, let's say, annotate the entire course. So I'm going to annotate everything just for the purpose of demonstration. So something like this. And this will essentially just save you a bunch of file. And then if you hold down Control-Shift on the keyboard and click in the stromal compartment, what QuPath will do is will make sure that those two different objects or annotations don't overlap. So wherever you have your epithelium, your stromal compartment won't go into that. So just to demonstrate how that looks, there. So I'm going to hold down Control-Shift and then I left-click on the stromal compartment. You see that it automatically aligns. So if I now double click on this object, it will be what I have specified as the tumor. When I double click on this object, it's a separate object that I will then set as true, right? So now I have my tumor over there and my stroma. And let's say that these two cores are from the same patients. So I want to analyze towards the end. I want to get the mean number of, let's say, cytotoxic CD8 positive cells in both these cores, but they're for one patient. So one way of doing it would obviously be to create my next annotation over here. Again, I'm just going to do this very roughly for purpose of demonstration. So again, this is the epithelial compartment. And then I'm going to erase parts that I identify as a stroma. So again, hold down Alt, click, and just brush away the stroma. Maybe same thing there, same thing there, and so on. Yeah, for example. Okay, and then I'm going to call this the tumoral compartment. So I'm going to set class tumor. And then I'm going to do the same thing for the stroma, right? So I'm going to fix this a bit. Yeah. So then I'm going to create another object. And I'm going to call this a stroma. So similar as to what I just showed you recently, I can do this manually and make sure that none of it overlaps and so on. But the easiest way of doing it is just highlighting all the parts that you deem as stroma, even if they overlap. And then you hold down Control and Shift and left click on the stroma, and it will make sure that those two annotations don't overlap with each other. So it looks something like this. So let's say I want to fix the epithelium to include that as well. Then I can go back and make sure that they don't overlap. Okay, so now I have, well, I'm just going to specify that this is the stroma as well. So remember, these two cores are from the same patient. Now, if I kept this separately like this, you have a total of 4 different annotations, right? 2 epithelial and 2 stromal. But if you then want to look at the mean number of CD8 positive cells, you would have to quantify the number here and quantify the number there, and then take the average of the two within an Excel sheet. That's quite tedious to have to do so. Another way of doing it would obviously be just to draw this annotation into this core and then erase everything that is a part of it. Or alternatively, as shown before, you can highlight this epithelial compartment and hold down Alt, select this epithelial compartment. So now you've highlighted both epithelial compartments that are from the same patient, and you can then merge those 2 different objects by picking up merge selected. So now after QuPath has processed that, if I select this object, it automatically highlights the epithelial compartment within both cores, right? And then I'll do the same thing within the stromal compartments. So I'm going to highlight the stromal compartment here and the stromal compartment there. And then I go into annotations, edit multiple, and merge selected. Now they're both merged. Now, like, let's say you accidentally merge 2 objects that shouldn't theoretically. So let's say you merge the stromal compartment of this core with the epithelial compartment of that core. Now you created 1 object. You don't necessarily want to have to delete your entire annotation and start all over again. You can actually select the newly merged object and reverse the step that you just did. So you can click on split. And now these 2 are split again. Right? So that's a pretty convenient way of doing it. But yes, this is how you can create your annotations. It's quite straightforward. And my recommendation is always to merge the objects, because then for downstream analysis, you won't have to calculate the mean manually in the Excel sheet. You just have this information for you readily available. So I'm going to merge the 2 objects again. So looking like this. But yes, these are the different tools that you can play around with. You can also look at the brightness of the image. The different channels. So you can look at the annotate channel by itself, and you can look at the DAB channel by itself. But yeah, my recommendation is basically just to play around with some of these tools and figure out the ones that you like the most. Good. So I'm going to go back onto my PowerPoint. Yes, and once you've manually annotated your objects, the next step, as mentioned, is to do the positive cell detection. So this is the next step. And again, here we're using CD8, a surface marker, to identify the cytotoxic T cells within the small bowel cancer cohort. Alexander, you will do something similar for the cohort that you're analyzing in your project as well. So we want to essentially use QuPath to identify how many of these cells there are per let's say millimeter squared. And QuPath is quite intuitive as a software to use for this purpose. So I'll show you exactly how you can do this. Let's go back into QuPath. Yes, so to do the positive cell detection, you have to highlight the compartment of your interest or your annotation of interest. So I'm going to highlight the epithelial compartments. Then again, you open up the command list, so Control L, and you type in positive cell detection. Or alternatively, it tells you also the path that you can use to find this. So it says analyze cell detection. You want to analyze cell detection, and you can do that way as well. But I just find it easier to use the command list. And then you double click on the positive cell detection. And these are the default settings that you have within QuPath. So you can try to use these, for example, to run it. But what happens most of the time is that you find that it's not perfect. And if anything, you play around with these parameters to identify the correct cells, because sometimes it will miss cells that are stained with brown, and those cells should actually be included in your analysis. Okay, so let's see, for example, here. The first thing you can play around with is the detection image. So if you have it set as hematoxylin, basically what QuPath will do is that it will detect the cells based on the hematoxylin blue stain, but also on the DAB brown stain. So just for the purpose of demonstration, I'll show you. It will detect the cells based on this. And this works quite well for a lot of the analysis that you'll be able to doing. The problem is if you have a cell or nuclei that's faintly blue. So let's say it's faintly blue, but the same cell has a very strong brown stain. So you know for a fact that you're quite sure that this cell is a cell. For some reason, QuPath is detecting that cell, and the reason it isn't doing though is because the blue stain is quite weak. So one way to get around that is you can use the optical density sum instead. And this doesn't necessarily detect the cells based on the hematoxylin stain. Instead, it just looks at the brightness of the object simply. So let's say I run it like this. And sometimes the results will be quite similar, sometimes they won't be. And again, the advantage of doing it this way is that you detect cells that have a faint blue stain, but they give off some kind of brightness. Whether that's blue or brown, it doesn't matter. It doesn't separate. It just looks for the brightness. And I can
Rammah, sorry,
yes?
just a second, because Alexander lost us. He got
Oh.
a black screen. It happened something strange to my screen also, but mine managed to couple back. So let's see if he's back.
Yes.
Alexander, can you hear?
Rammah, in the meantime, I have a question. And
yes.
Measure some spheroid, but I use ImageJ. Is possible to with QuPath also measure a structure like that? Because yeah, in ImageJ, in the other program, I have to select the area of the spheroids, and then I will have what is the behavior during the time, right? In QuPath also I can do this kind of things.
But what is you're probing or querying within ImageJ? Are you looking for the number of cells within the spheroid?
No, it's just the area. See
area.
how they grow. The size.
Yeah. Area will be like you could do that definitely, because you create an annotation within, because you see your image, right? And then you create an annotation for the spheroid, and then within that annotation you give a whole bunch of parameters, including the area.
Yeah, but my question is at the beginning, for example, in ImageJ, I need to measure first the scale. And when I have the media of this scale, I put these settings in all the global image setup, and I can get the area. In QuPath, how shall I start doing that? Any idea? But I can continue in ImageJ, it's just curiosity.
I think in QuPath will be according to the magnification the tissues were, isn't it, were scanned.
Yeah.
Because Lorena has pictures from the microscope.
From a normal microscope.
It doesn't have, from the
normal
inverted. It's inverted.
Okay, I see. Yes. Yeah, so then I think I would agree with that, and it has to do with magnification that you're working with. But I think maybe if you want, Lorena, you can show me what you do in ImageJ, and I can see if I can do something similar with the QuPath.
Yeah, I can show you.
Alexander, are you back? We don't hear you because you're mute. Let's see if I can unmute you. Hello. He is there, but I don't know. Actually, I think I can
open the cell detection at the.
So here?
Yeah. So when you first open the thing.
Okay. Yes, no problem. Yes. So then I'll just continue on from there. I guess you saw how I opened positive cell detection, but again, it's just simply Control L, open the command list, then you type in positive, or alternatively, you can also type in cell detection. Then you select it, and automatically open up the window where you specify the parameters that you want to use for the cell detection. And what I mentioned recently was the first parameter that you have to adjust is the detection image. So here you have two options. The first is hematoxylin OD. The other is optical density sum. If you use hematoxylin OD, the detection is made based on the hematoxylin stain within the image. So I'll just show you how that looks. Just 1 second. So it's basically done based on off of this. So this works quite well most of the time. The problem is if you have a cell that has a very faint, weak hematoxylin stain, then QuPath will identify that cell, or won't necessarily pick up that cell in the positive cell detection. So let's say you find a cell, there's a very weak or faint blue stain, but there's a very powerful or strong brown stain. So you think, or you're quite sure that it is a cytotoxic T cell or CD8 positive T cell, but QuPath has for some reason not picked it up. And this is likely due to the fact that the hematoxylin stain is quite weak. So a way of getting around this is changing the detection parameter to optical density sum and running that instead. So it looks something like this. And basically what this does is it then detects the cell based on the brightness. It doesn't use the hematoxylin or the DAB channels. It doesn't care how blue or brown these cells are separately. The only thing it cares about is how bright is the object that you're trying to query. And this works quite well also a lot of the times. Where this becomes an issue is if, let's say, you have a core that has a lot of artifacts. So let's say for some reason during the processing of the TMA or the processing of the slide, some parts of the stroma have been, I don't know, washed away, or you see a lot of artifacts. Then because of the fact that here you're picking up object space off of the brightness only and not the hematoxylin with the DAB stains, you could potentially have a lot of false positives as well. Pick up those artifacts in your analysis. So what I would suggest is when you do your analysis, try to play around with both and see which one work best for you, depending on how your TMA looks. But now I'm just going to go back here. Run it like this. The second parameter that you can play around with is the pixel size. So generally the concept is if you reduce the pixel size, and you do so according to the pixel width and height that are, or the pixel width and height that are provided here in this left panel. So here the requested pixel size is 0.5, but the pixel width and the pixel height is 0.22, approximately 0.25. So it's essentially a factor of 2. So when you then run this analysis, you'll have to downsize by that same factor. So if you do this, it could potentially improve your analysis. The problem is the more you reduce it, the more the analysis takes time. And can potentially also crash QuPath. And sometimes, no matter how low you go, it won't even be a big difference. So let's say here, the positive percentage is 0.5%, right? So this was for 0.5. If I do it for 0.2, let's see if that number changes. This is what I get right now. .5 still. So again, as you see, the difference isn't the biggest, right? So I usually just keep this at 0.5, but you can try to play around with it and see if it makes a big difference. And if it does, you might want to consider adjusting this parameter. You can also go the other way. You could increase the requested pixel size to 1, 2, 3, etc. That will make your analysis go a bit quicker, but at the same time, it might affect the performance of the analysis. So yeah, you can try playing on these two parameters.
I think it detected too many nuclei when you had 0.2. So for example,
did it? Yeah.
Yeah, was not accurate. So
no.
Alexander, you need to look also how these parameters will be reflected in the way the cells are segmented. Yes. And if you see some unusual segmentation, then it's not good to go too fine.
No.
And it will detect false cells where they are not. But also, when you have a lot of inflammation, then you might have a lot of cells together, and then you could use this, at least from my experience. But for him, for P53, he will just analyze the tumor, the epithelial compartment. So then I think he doesn't need to go to a very low pixel size. So 0.5 or even 0.6 would be okay, I guess.
Yes.
Sorry.
Yes. Yes. No, very good. Yeah, I found during my project that I had to play around with this parameter a bit myself, but OpenQuive seemed to perform quite well for me when I was doing my analysis. And yes, the next parameter that you can play around with is the background radius. So basically, what QuPath does when it does positive cell detection is that it first subtracts the background, and then it applies the threshold to identify and detect what's a positive cell and what isn't a positive cell. So when you play around with this parameter, you also need to keep a lookout for this parameter, because let's say you have a very high threshold. Then if you have a very high threshold, what will happen is that QuPath, let me see if I can try to demonstrate it. Let's see. No. So it won't detect anything basically, because you've set your threshold too high, and this is based on the MFOX on the channel. So if I reduce this, it should go back to detecting some of my cells. I can reduce it even more. There, and then I can detect more of my cells. So this again has to do with how your cells are. But basically, if you have a very high threshold, then you don't necessarily need to have a background subtraction, because you already have a high threshold. But if you have a low threshold for your antioxidant, then you might want to introduce a background, because then otherwise you end up getting false positives. So again, if you play around with this parameter, you also need to potentially adjust this parameter as well. Yes, and then when it comes to the median filter radius and the sigma, this has to do with the fragmentation of the cells. So I'll demonstrate, for example, I'm just gonna show here, if I increase it to 3. What will happen is that, like, let's say these cells over here, it will merge a bunch of those cells together, when in reality, you think that those cells should be separated. They are separated cells, but they've been merged together by QuPath because you put in a too high median filter radius or a sigma parameter. So I can do the same thing with sigma. Let's say I put it as 4. And then it will also start just putting together cells that really shouldn't be put together, right? They should be separated. And the vice versa is also true. So if you have these too low, what will happen is that it'll essentially start fragmenting cells that shouldn't necessarily be fragmented. It just become a bit chaotic and do something like this. So again, play around with these parameters and try to make it look the best possible way within your analysis. But essentially, you don't want the cells to be too merged together. You don't want it to be too fragmented as well. So you just have to play around with these parameters and figure out what's best within your TMA, within your image. So I'm just going to use this for demonstration right now.
But Rammah, from your experience, did you have to play a lot with these parameters or not? From my experience, we didn't have to play too much. Maybe.
No. I did. In my experience, I played around with it too much and realized that I shouldn't have done it to begin with. You know what I mean? For me, it's just more about learning what everything does. But again, when you look at your TMAs, I think what you'll see is that if you notice that something is very wrong, then you can maybe start making small adjustments to one parameter at a time, as opposed to making a whole bunch of adjustments, because that thing just becomes chaotic. If anything, like Dana said, I feel like the standard parameters they work quite well. And then you look at your TMA, and if you see that something needs a small adjustment, you can then adjust that specific parameter one step at a time. So one parameter at a time, basically, is what works best for me. Yes. And the minimum and maximum area simply have to do with the size of your cells. So let's say if you put this as 1 or 2 micrometers squared, it could also potentially end up detecting artifacts, so things that aren't necessarily cells, as positive. But again, here you might not have to change too much within your DNA, because these numbers might work very well for you. And vice versa, if you end up detecting cells or if you end up missing cells that are quite big in your TMA, you can also adjust the maximum area of these parameters if you need to, but you don't necessarily have to. The threshold parameter I already discussed. It has to do with the detection of the cells based on the antioxidant channel. So if you have a very high threshold, then it will detect anything from earlier. If you set a very low threshold, then even things that are very faintly blue will also be detected in your analysis. Something like that. The split by shape also has to do with the fragmentation, but essentially what it does is it tries to identify cells as circular objects. So let's say you have a structure that is very not circular, then those two objects might be merged together. But within my analysis, I tend to just kind of keep this one, and it seems to work quite well for me. The exclude dab staining has to do with, let's say you're looking at a surface marker, and you only want to look at that in the what's it called again, in the cell membrane, but for some reason it isn't able to do that property, and it sees it in the nucleus. You could then potentially try to toggle this and see if you can fix that problem. But again, for my analysis, I've never really had to do so. So I think you just kind of keep this off what you're doing, Alexander. The cell expansion has to do with, so let's say this cell over here, it has to do with the radius around the cell that's being also used to detect whether that cell is positive or not. So I can potentially change this to, let's say 1 and 1 again. And you see that this radius should decrease, should become smaller, something like that. And you could increase it to 5. So the problem is if you increase it too much, it could potentially, let's say you have two cells next to each other. One is brown, the other one isn't brown. Then the cell that isn't brown, if you have the radius too large, it could potentially include the brown staining of the other cell that is brown. So we'll identify a cell that isn't actually positive for CD8 as positive only because of the fact that you have a radius that's too big. So you need to keep a lookout for that as well. And the final parameter that I want to discuss is just the threshold, the intensity threshold parameters, and this has to do with dab staining. So let's see if I can find some here. If you set this as too high, let's say threshold as 3, then it's going to identify the cell as a cell, because again, as mentioned, this is based on toxicity, right? But it won't identify it as a positive cell, because the positive cell detection is based on the dab staining. And we see for a fact that there is dab here, so this does to me look like a positive cell, but we put our threshold too high. So for that purpose, I'll have to reduce it to maybe, let's say 0.8, and we run the analysis. And then that didn't either. So let's see. Let's try 0.08. And now the cell is picked up. But the problem here is now picking up something that shouldn't be picked up. So maybe I want to have a higher threshold, or alternatively, because this looks like it's not very strongly stained with hematoxylin, you can play around with the parameters for the hematoxylin to try to eliminate that cell, but to keep this cell as a positive, as positively stained, basically. So yeah, basically when you run your analysis, you're gonna have to the parameters that I did have to actually adjust, not a lot, but adjust to a certain degree, are the parameters for the dab staining and the parameters for the hematoxylin. And this is basically to make sure that you kind of remove all things that shouldn't be necessarily positive detected, but make sure that the ones that are positive are actually detected in your analysis. And Alexander, I will mention that it'll never be perfect, but you just try to make it as good as possible and make sure things that aren't glaringly incorrect, you know, for example, this or that, those things shouldn't really be included in your analysis. Yes. But just so once you run your analysis, then in the left panel here, you get the information, the output of the positive cell detection, and it'll give you the number of detections. So this is the total of number of cells. They give you the number negative, so the ones that aren't positively stained for CD8, number positive, the percentage positive, and the number of positive per square area, per millimeter squared. And this will then depend on what you're interested in for your analysis. So you can then transfer all this information to the Excel sheet for downstream analysis.
Rammah, can you show him how he can switch between the segmented cells, the image with segmented cells and non-segmented cells? So when he will evaluate if his algorithm is good or not.
Well, what do you mean, Dara? Sorry?
Well, you have this, that it's this app that you can click on to see the segmented cells and non-segmented cells, the image with the border of the cells and without.
Without the border of the cells?
Yeah.
Ah, you mean
yeah, just that.
Okay.
Yeah,
I see what you mean. Okay, yes. Okay, yes. Yeah, so you just toggle this button.
Because this is useful when he modifies this to just
very, yes, definitely very. So yeah, because otherwise it gets very messy if you look at it like this. So it's useful to, as Dana mentioned, be able to toggle on and off between this to see if it looks correct or not for you. And you can also fill them in like that with this button here. Good. So now I'm just going to run the detection here, and I'll run it here. So it looks okay. 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 Yes. So now we'll run the positive cell detection. Just going to go back onto my slides. And the final step after you've run your positive cell detection is to transfer all of that data onto an Excel sheet. So it's this part over here, and that's quite straightforward to do. So just 1 second, close this. Yes, so basically what I want to fix is that I forgot to merge all of these objects together into the stroma compartment. So I'm just going to do so quickly because this is all part of the same core, right? This is stroma from the same core. So as mentioned before, I can double click and select the stroma compartment, hold down Alt, and then select all the different parts that should be part of this annotation. And then I can go to edit multiple and merge selected. And now all of them are selected. So this looks more correct. And then I'll do the same thing here. We're selected. And that looks correct to me as well. Good. And then I'm going to merge the two stroma compartments from the two different cores. That looks like it's already been done. Yes. Okay. Now I'm happy with everything. Basically, I just want to make sure that I have two annotations for the two cores for the stroma and the two. So now I can run the detection again, just to make sure that all the numbers are updated for what I just did. And yes, so you'll have all the information as mentioned in the left panel over here. But if you then want to transfer that information onto an Excel file, you basically click on this grid toggle over here, which says Show Measurements Table. And you click on Show Annotation Measurements. And then you can click on Copy to Clipboard. And these are basically all the parameters that you can copy onto your Excel file. And then you can just create a workbook, and then you can just paste that information. And you'll get all the information that you need then for your downstream analysis. So once you've done it for all of your cores, you will essentially just get rows and rows and rows for all of your cores. And one important thing that I do need to include as well that I haven't included so far is because, again, all of your cores have identifiers, right? They have numbers or letters. So let's say I'm gonna call this core patient A. So you can do that by doing annotations, set properties, and I'll call it patient A. Apply. And then I'm going to do the same thing for the tumor compartment or epithelial compartments. Patient A. And the way to do so is to go into annotations and set properties. You can change the name of those. Okay. And then again, I'm going to copy this information. I just want to show you the difference. The important thing is I now have the name of my annotation as well. And why this is important is because when you then have the layout for your TMA, you can then match this information to the specific cores that are in your layout as well, right? So you know which patients have which numbers, and then that will help you in your downstream analysis as well. So that's quite straightforward. And the last part of this presentation is just to show you something that you might not necessarily be doing, but it's just interesting to know. And that's basically the machine learning approach of separating between the epithelial and stromal compartments. So this is something that I've been playing around with a bit myself, but I haven't managed to optimize or to do it perfectly, at least enough for it to be included in my reports, but maybe it's something you can try out as well, Alexander. So I'm just going to go into the other image. There. And I'm going to rotate it. Right. So the reason I want to do this is because, sorry, let's rotate it some more. 1 second. Yeah, that's how I should look. Yes. So the reason I want to do this is because, as you can imagine, Lysander, like to manually annotate each epithelial and stromal compartment, for this entire TMA can be quite tedious. It can be quite time consuming. And what people are trying to do instead is to use a machine learning approach to essentially select all of these different cores within the tissue microarray, within the TMA, and then to use the object classifier within QuPath to separate between the epithelial and stromal compartments. Because if you were able to do this successfully, then maybe 2 hours of work could produce the same results as like days and weeks and months of work, to be honest. But again, when I've been trying to do this as perfect, so it is something that you need to maybe play around with yourself. But the first step is to do something called the de-arrayer. So you click on the TMA tab over there, and then click on TMA de-arrayer. And this first parameter here has to do with the core diameter. So each of your diameter cores, and you can adjust this afterwards if you're not happy with it. So I'm just going to ignore it for now. You then need to count the number of columns and rows that you have. So here I have 1, 2, 3, 4, 5, 6, 7, 8, 12. And so I'm going to put that there. And then I have A, B, C, D, E, F, G, H. So I have A to H. And then I'm going to specify column first. And then you can run this. And basically, it's supposed to select all of the cores in your TMA. So it looks something like this. And let's say I'm not happy with the size of this because there are parts of the core that are not being included in my analysis. I can then increase the TMA core diameter, and I'll make that circle a bit bigger. So this looks a bit better to me, but if you wanted to, you could also potentially increase it even more. So maybe something like this. No, it's too big. But yeah, anyway, so we can play around with that. And then once you've created your TMA array, you then do the same thing as we did earlier, which is to estimate the staining vectors. So we're going to use going to Control L, my command list again, then write stain vectors. And then I first have to create the object like I did last time. So I want to have some background. I want to have some tumor cells, some blue cells, and some brown cells. So maybe something like this. And then I do estimate stain vectors. And because I've included the background, I select yes here. I'm going to use the auto function to do it automatically. Again, same as I did last time. So it changes the stain vectors. And then click on OK. And then once those have been updated so that they're correct, I can then delete this annotation because I no longer need it. The next step is to then run a positive cell detection. So if I was to do this for this entire TMA, that would probably take minutes, and it can also potentially crash my QuPath. So I won't do that right now. I'm just going to select a couple of them, and then I'm going to run the positive cell detection. So for the purpose of demonstration, I'm just going to run it using most of the same default parameters. And then I click on run. This will take some time, but it won't take too long time. And again, so far, I haven't separated between the epithelial and stromal compartments, right? I'm just kind of running the positive cell detection for the entire course. And hopefully, we'll be able to do some of it. So then it'll look something like this. So as Dana said, you can toggle on and off to see the cells and to see the positive cells. But as mentioned so far, it's just the total number of positive cells in the entire core, and not necessarily the epithelial or stromal cells separated, right? But that's what we want to do. We want to separate the two without having to manually annotate it. And the way you can do that is to use the machine learning algorithm in QuPath, and it's called an object classifier. So you would select train object classifier, and then you basically before you start doing the training, you have to tell QuPath, you have to feed information into the software and tell it what's epithelial compartment and what's stromal compartment. So let's say here I'm going to create an annotation that looks something like this, and I want it to know that this is the epithelial compartments. But here, this is the stromal compartments. And then I'll do the same thing here. This is epithelium. And then this is stromal. Okay. And then I should be able to run this. And then if I look at it now, it will automatically separate between the two. So remember, the stroma is highlighted in green, the epithelium is highlighted in blue. So as mentioned before, usually you would have to manually annotate these by yourself, right? You'd have to select all of these objects and say that this is epithelium and that is stroma. The advantage here is that it does that for you automatically. And where this makes a really big difference is, let's say you have a core like this. So to annotate this manually is a headache because you have to draw all of these little tumor islands. You have to make sure that you don't do the stromal compartment to the epithelial compartments. You have to erase, and doing a core like this can take, 10 minutes. It can take quite a lot of time actually. So the benefit of using the machine learning algorithm is that you can potentially get around that because this will do it for you automatically. The problem becomes is that it then ends up identifying certain objects as for example, stroma, where if you had done this manually, you would have annotated most of this as epithelium. So to improve the machine learning algorithm, you essentially have to feed more information into it. So you have to keep drawing objects and specify their class. And then the algorithm by itself should update and try to improve the separation between the epithelial and stromal compartments. But as mentioned before, I did this, but I still struggled with it to a certain degree to make it perfect, especially when you have, let's say, necrotic areas within your TMA or within your pores. Those necrotic areas might be picked up as the incorrect compartment. So this is something that you might have to play around with to try to optimize, but if you can make it work, that would be great because you'd save a lot of time. But just, this is a possibility with
Rammah, I have a question.
Yes.
So
Yes.
when, let's say, because I think for P53, he can do this, because P53 will be hopefully just in the cancer cells. And then how he can name the cores to get, how do you think?
I think they're automatically named because, let's see here, it's 3A. So it has to do with the orientation of the DNA. So
no, but I mean, then he has to still name each core.
I don't think so. Let me see. I'm just going to try to copy the information. Second. Let's see. Okay. I was hoping that it would be named automatically because it is strange. Okay.
But maybe we'll figure out.
Yeah, maybe I can look into that myself as well. Because it's strange because here in this panel, you see that there is a name. So it has given some kind of name to it, and it kind of makes sense based on the column and row orientation of it. So column 3, row A, right? That makes sense. But for some reason, when I transfer that data onto Excel, it doesn't show up. So
it doesn't keep it. Maybe
no. But there'll be an easy solution for that, I'm sure. I can look into that and get back to you. That shouldn't be a problem. That should be straightforward.
Me and Alexander, we can look at it tomorrow and then maybe come back to you.
Yes. Yes.
Okay. Sorry for the interruption. Yeah.
No problem. I think that's actually it. But yeah, what I wanted to say finally is that if you wanted further information about this, about the QuPath, you can go on YouTube and Peter Bankhead has a pretty good tutorial on how to use it. So on YouTube, you can follow that tutorial. It is about old. It is a bit outdated. QuPath has been updated quite a lot since then, but I think it's still a very good way to start when you want to get into it. And he's also done a recent presentation just giving background information about the software that you can also access. And he also has these beta doc files that kind of explain the different steps or the different functions that you have in QuPath. So these are some of the resources that you can look into Alexander that might help you to essentially start what you're going to do at QuPath. But yeah, if you have any questions while you're doing your analysis, obviously just let me know and I can try to do my best to. Yeah, do you have any questions?
Thank you. I will stop the recording now.
Yes.